So, students, before sir join us, let me give you a brief introduction about him. So, I'm not going to take much of his time later. So, guys, very good morning, everyone. And we have with us Dr. Pranjal Chandra. He is Assistant Professor and Ramanujan Fellow at School of Biomedical Engineering, IIT BHU, and his field of specialization is biochemical sensors, nanobiosensors, commercially viable diagnostic platforms, and nanobiotechnology. He did his PhD from Busan National University, South Korea, and postdoc from Technion, Israel. He has published 10 books and over 100 publications. There are four PhD graduates graduated from under his guidance, and there are several master students are working under him. And uh, uh, he is also associated editor of Census International and guest editor of Molecules, board member of several other journals. He is, recipient of many awards. Like he was listed in the top 10, 2% scientists in the world, and the list was prepared by Stanford University, US in 2020. His team has received Gandhian Young Technology Innovation Award in 2020. He is also a recipient of Brain Korea 21 Fellowship, South Korea, and National Research Fellowship from the same country. He is recipient of Ramanujan Fellowship from Department of Science and Technology, as I have mentioned. Early Career Research Award from DST, and he is also a doctor, received award from Dr. OPS Sensors. Yes, sir. The slides are visible.
All right. Yeah. Good.
Yeah, and he's also a recipient of Young Scientist Award 2017 from Biotech Research Society of India, Young Engineer Award in 2018, Young Scientist Award in 2019, and highly cited corresponding author in the Royal Society of Chemistry. Cambridge, London, in top 10% cited articles in Journal Chemistry section. He's also selected as a member of National Academy of Science in 2020, and Outstanding Faculty Award in terms of teaching and research. So, with this brief introduction, on behalf of Srishti, I welcome you, sir. Once again, thank you so much for accepting our invitation for this talk.
Thank you, Dr. Megha, for a very generous introduction. So I'm sure that my slides are visible.
Yes, sir.
Right? So,
sir, can you do slideshow?
Yeah, sure. Is it fine?
Yes, sir.
Okay, all right. So, thank you, Dr. Megha, for a very generous introduction. And I'm very thankful to Srishti once again, because I was in that 6th summer school also. I gave a talk, and of course the Srishti organization and DBT. I'm very much thankful for BIRAC, of course. So giving this opportunity to discuss some of the work what we are doing in the lab, and not only in our lab, and what are the trends basically going on in the respective area. So this is actually very much encouraging for the students, and not only for students, for the people like us who are trying to connect the science for the society. Okay, so with these few words, I would like to come up to the point what we are going to discuss today. So the topic what I'm going to talk is paper-based sensing technologies, design and applications. So as Madam has said, that I'm working in the Indian Institute of Technology, BHU, Varanasi, and whatever we have done, it has been done in my lab. And in addition to that, I'll be talking the most recent trends and how the system has been integrated for various applications in the next about 45 to 50 minutes. So I would like to mention that the students who have participated in this presentation, so I've seen their background also. I mean, the Excel sheet has been shared by the Srishti, and it is very good actually, so that the speaker should know the student from various part of the country they are participating, and which grade they are studying. So accordingly, we have to we are getting a chance to tune our presentation based on that. So the contents what I will be talking is what we study at IIT BHU Varanasi, and the challenges in the medical diagnostic and healthcare systems, and how this diagnostics and healthcare system can be relate with the paper-based diagnostic. That was the point of discussion today, and different type of paper-based biosensors has been developed and how they are going to integrate with the molecular diagnostic technologies. All right, so this is my lab, the laboratory biophysics sensors and nanobioengineering. So this is the overall things what we do in our lab. It's a material engineering and characterization, development of commercially viable systems, medical diagnostics, nanobioengineering, electrochemical sensors, and of course, paper microfluidics and biosensors. I've highlighted this explicitly because we are going to talk only on this point in today's presentation. So basically, when we talk about any kind of the system, what we develop, either a paper or a conventional system, we should know that what we are studying basically. So we are studying the detection, right? So in either you use a paper or any other system. So major idea is to detect a molecule of interest. So that is the major thought in the mind. So I have given for some examples here. The most common nowadays people are listening is a COVID-19 antigen. So I mean, you will be surprised to know that even the paper-based diagnostic system has been utilized for COVID-19 antigen testing also. So in addition to that, COVID-19, you can go for a whole cell. Either you can go for a toxin, you can go for a DNA, you can go for RNA or any protein level. So the basic thing what I'm trying to explain here is what. So basic is the biomarker is very important for any biosensing system, right? And if you detect a biomarker, eventually you're going to diagnose a disease. Okay, so these are the conventional techniques what is listed here in the pictures. So these are very powerful system, right? But the problem sometimes it is very laborious and it requires very large sample volumes and a time consuming protocol and so on. So the basic idea is what we can integrate a new technology, we can develop a new technology, and the major challenges in the case is what? It's a quick detection, on-site detection, and ultra-sensitive detection. So these are the challenges as I mentioned. So this we need to overcome with the smart sensing approaches, which is one of the very promising approaches nowadays. So the answer for this system is what? It's a biosensing system. Okay. So this is basically the pictorial representation of the biosensing complete layout. And there are two major things. What is important in this case is what is the biosensor and the transducer system. So biosensor is basically an analytical device which integrate the bioreceptor with the external sample here in this side. So in this red color dotted line, these are the biorecognition element. It could be a DNA, it could be a protein, it could be antibody, or and so on. Many other receptor is also possible. And then there is a transducer system, and then it amplifies and you get output data. So output data is very much common in case of the optical signal, mechanical system or electrochemical system. So if I talk with reference to the paper-based system, so majorly the output data is either optical or electrochemical system. So that we will see in the coming slides. Okay, so these are the basic introduction of the system, which is very much important to understand. Okay, so I'm coming a little more scientifically here, that the conventional electrode system what we use in the lab is. So these are the website you can go directly and you can see these images. So I have taken this pH meter also because I'm sure that the student in BTech or MTech they are using the pH meter always in the lab. So this is also one type of electrode. This is an ion selective electrode basically. And this is the conventional electrode, which is called as a glassy carbon electrode, right? It could be of various length, various diameter, and this is another type of micro electrode. This is a gauge type of electrode. So these are the reference of this particular information. And then if you see the pictorial cartoon of the glassy carbon electrode, it's a electrical wire, there is a rubber ring, and there is a Teflon tube, and there is a composite system what you can use onto the surface. This black area is basically responsible for the whole sensing process. All right. So another is what? It's a pH electrode. So this is ion selective electrode. What I'm trying to discuss in this slide is what these are the conventional type of electrodes what people are using. In addition to this cylindrical electrodes, there are planar electrodes also. So this is one company which sell this kind of electrode. These are called as a screen printed electrode basically. These are small chips basically. So I'm sure that everybody who has seen various developments in case of the glucometer in the market, they use particularly this type of small chips. So this is the conventional system basically, what is important to diagnose any kind of the molecule. So based on these systems, the conventional approach for the detection is what is an electrochemical system. And this is a conventional setup where there is a 3 electrode is there. One is a working electrode, one is a counter electrode, one is a reference electrode. And you do a lot of reaction here and ultimately you get a signal output here. All right. So this is not the point of discussion what we are going to talk in terms of the electrical signals basically, because we are focusing on a paper-based system. But yes, the electrochemical readout can be integrated with the paper-based system also. Okay. So different prototypes for the paper-based biosensor which is present nowadays is one is the lab on a chip system. The word itself says lab on a chip. It means whatever system we are trying to do, either the concentration, pre-concentration, or the separation, so that can be done on chip basically, one chip. Another is a micropad. It's a microfluidic paper-based analytical devices. So this is one of the example of that. If you have a contaminants in the water, you can put in the multiplexing system. Multiplexing means one system you can go for a multiple sample analysis, and then you can integrate with the mobile phone, or you can do some other processing, and eventually you can have the data analysis in this case, right? So the most common system which is in the market is basically the lateral flow devices. So if you see any small device based on the paper system is a lateral flow device. For example, the pregnancy strip, for example, the malaria testing kit, something like that. So this is very much common actually in the market. And the most recent one is what the Abbott has developed a recent system based on the paper. That's a COVID-19 antigen card. So what I'm trying to emphasize here is what the paper can be very widely used for various type of diagnostics, and in that case there are various versions of that basically. One is a lab on a chip system, one is a micropad, another is a lateral flow system. So this will give you the the picture of basically what is happening across the globe and how the paper is going to be benchmark kind of a system for in case of the diagnostics. All right, so as I said earlier, so there could be a possibility that 2 different readout can be integrated. But if you see the previous slide, this one, in most of the cases, for example, if this one or if you see this one, the output signal is an optical signal. I mean, it means the color will be changed. If you see the conventional malaria testing kit or pregnancy testing kit, the paper system what has been used, you'll see the line, the red line, double line or single line, and you will be interpreting the results. Such example has been shown in this point basically. Okay. So what I'm trying to show, this is optical readout system. However, whether the system can be integrated, for example, the electrochemical can be integrated with the optical, or paper can be integrated with the other system where you can get electrical output. So this is the various examples. Like in analytical chemistry, the paper matrix has been utilized to develop various electrodes that have been printed onto that, right? And these are the dimensions of that. And with this one, they have developed what a multiplexing system. For example, the glucose, the lactates, and the uric acid, they can be detected simultaneously using the paper-based platform. So that is what I'm trying to convey here. And another, this is the cascade system where the paper matrix has been utilized to develop the diagnostic kit. Okay, and this is another example where the paper has been coated with various samples, and eventually the working reference and the various electrodes has been developed onto the paper matrix, which is the conventional system in the electrochemical system, right? And in 2021, I mean last month in January, we got a paper which has been published recently in Electrochemical Acta. It can be this is very important basically, because paper-based electrochemical platform here has been developed, and various nanostructure material has been utilized for the sensing of dopamine, which is a very important neurotransmitter. And here you can see The signal is in terms of the current, nanoampere. So what I'm trying to explain here is what the optical as well as the electrical signal can be obtained by paper-based system. So this is the summary of various paper-based electrochemical sensor which has been developed so far. So I've also cited the reference if some of the students they want to go and read the paper in detail. So the electrode fabrication method, which is very much common nowadays and very much important, is basically the inkjet printing. I mean, it's a simple scanning system. Like if you have a printing system in your house, that can also be used for that. And of course, there are various advanced systems also which can be integrated with that kind of a printing system. So this is normal screen printing system. It's an inkjet printing system, inkjet. This one. So this is the microfluidic fabrication method they have been followed. This is the type of the ink what has been used. In addition to the normal system, they have utilized the nanomaterials also. This is the electrode modification. It means, as I said earlier, they have various receptors. For example, in this case, if you want to go for a glucose analysis, you need to have a glucose oxidase onto the system, right? So this is the receptor system, and this is the limit of detection of this particular system what has been developed. Okay. So why paper-based system is important for personalized healthcare? Because there are basically P4 system. One is the predictive, preventive, participatory approaches, which is very much important. And eventually, when you develop this one, it goes for the personalized diagnosis. I mean, that small paper-based device, definitely you can keep it in your pocket, and you can go for on-site analysis. That is the advantage of the P4 system. And for the P4 system, the most important thing is what the paper-based device is very widely used for the P4 analysis. Okay, so these are the various details what I've mentioned on the slides. You can read it later on. So now, the most important design criteria when you go for the designing of the system, either a paper sensor or any diagnostic system, this is extremely important concept because, I mean, I have discussed with reference to the glucose level in the body. So in the left side, this side, you can see this is a low glucose sugar and this is a high glucose sugar. So what doctor is interested to know is what is the range, basically, what is the concentration of the glucose. Only then he can give you any medicine. So of course, when we design the system based on the paper matrix, we also consider this thought. For example, I've shown in the previous slide that you have developed a glucose sensor, and then glucose oxidase has been loaded onto the paper and so on. But in that case, the range of analysis should be very wide, yes, right? It means if some of the sensor is very good and it is able to detect the glucose from 80 to, for example, 120 or 140 mg per decil glucose. So it is very narrow basically. So what is the point? The point is basically it should be as wide as possible. If you see this one, it should go from at least 70 to, for example, 450 if it is a very hyperglycemic condition. So this range is very much important. And whenever we report the analytical details of any paper-based sensor, we need to report that LOD, what is LOD? That is called as a limit of detection. The LOQ, what is the limit of quantification? LOL is the limit of linearity. And dynamic range, it means with concentration increase, how much the signal is keep on increasing with a very good R-square values. It's a regression coefficient, and you have to do the regression analysis in that case also. So basically, this gives the slope of this plot gives the sensitivity analysis, and based on this values, even the numerical values what you obtain for any device, we will be knowing that which sensing system or which sensing readout is very much applicable for point of care analysis, and it is going to help the public as a whole. So this is what I want to explain from this slide, that everything is a level and the sensor should comply these particular points. So there is one more thing. If you read a lot of literature in the given, so in most of the cases, they are using a lot of nanomaterials in the system. So what nanomaterial does basically, when you add a nanomaterial in the system, it eventually, increase the slope of the system. If you go back to this slide, the value at this point at a particular concentration will be higher. This is one thing, right? That means improve the detection range, and it stabilizes the system, and it provides a robust system because, as you know, the nanomaterial has a very important, very unique properties in terms of its electrical property, magnetic property, and so on. And then it also increases the biocompatibility based on the matrix what you're using. And it also helps the bioreceptor immobilization. For example, if you take various nanostructure material, for example, the graphene or carbon nanotubes or any other carbon-based material which has a functional group. So that functional group can be, integrated for immobilization of various bioreceptors such as the antibody or aptamer or enzyme. The classical example is the reaction between the carboxylic acid and NH2, which gives rise to the CONH bond. So this is a covalent bond which is very much stable onto the system. So eventually the sensitivity will be increasing when you are integrating the nanomaterial into the matrix, and then the signal to noise ratio also increases. So overall the performance of the system increases when you involve various nanostructure material in the matrix. So the scientific trends over the years is what basically from 1997 until 2020, I have not added the data of 2021. It is keep on increasing if you see that. So every year there is the number of publication is keep on increasing. This shows the importance of the paper-based matrix, and we should also remember that the first example was for the paper-based system for bioanalytical application was shown by Martin and Sings, and for that purpose they got a Nobel Prize in 1952, right? So and this one is the recent publications in terms of the books basically, and the most recent book has been edited by Professor Urban Marcosi, and it has been published by elsewhere. So this shows that the importance of paper-based diagnosis system is very much important, and it is coming very largely over the years. So this is some of the, in addition to the academic based system, you can see the market potential also. This is the survey of 2018, which shows that the paper diagnostic market is going to be this amount in 2025. And the most recent, last year, it says that it is going to be 10.69 billion by 2027. So this clearly shows that not only the academics, but the commercial point of view of the paper's diagnostics is very, very much important. You go to any medical shop, you ask for a device, malaria testing kit, or even the COVID-19 testing kit, or any device, even the glucometer, you will get a paper-based prototype. So that is very, very much important. It shows that it has a very high demand in case of the diagnostics, right? So the commercially available systems, as I have mentioned many times, you can see the malaria testing kit, the drug testing kit, the sugar testing kit, COVID-19 antigen testing kit, and even HIV testing kit is present in the market, right? So that says the importance of the paper-based matrix in case of the biosensing system. So if you see all these slides, in particular the drug, malaria, HIV, and COVID, they go for optical analysis. However, in this case, you can get the digital response, right? So that's what I mentioned in the previous slide also. It go with either a optical system or it go with the electrochemical system. So that is the importance, and this is the the uniqueness of paper-based matrix. Okay, so I'll give you now the examples of the various type of paper-based biosensor. This is a dipstick-based paper-based sensor. This is one of the type. Basically, I'm going to discuss the type. So dipstick, the word itself is very clear. You are dipping this kind of a stick like a pH strip, right, into the solution. You are going to get the change in the color. So that kind of the system is the basic idea of the paper-based diagnostics. And on this, in this side, you can see that nitrite, protein, pH, blood, and specific gravity, that can be, tested by the dipstick-based technique where the paper has been utilized in a matrix. And this is one of the classical example of the uric acid detection methods, where also there is a change in the color, and then that can be integrated with the calibration system for the uric acid analysis. The detail of this particular work has been given in this reference, right? So in this one, this is one of the type dipstick type paper-based biosensor. This is another system which is called as a lateral flow assay. The difference in case of the dipstick and the lateral flow is what? Basically, it offers a controlled fluid flow, right? That is a little, advanced system compared to the dipstick system. And this is a very meticulously designed matrix. right? You have a dimension sets, you have a various region of, the movement of the solution, for example, a urine or any sample. So basically, if I go it in a little more detail, there are various positions in a typical, lateral flow assay. One is the sample pad, this part. One is the conjugation pad, this part. It means you're going to have some conjugation reaction here. It means the complexation reaction. The detection pad, this part, and absorbent pad, this part, right? So these are the, the information which is very much important. But the most important point is what you need to have a conjugation pad where you will be doing the conjugation reaction. And after loading the sample here, for example, this is for a PSA. I mean, this is one of the examples for the lateral flow assay. When you're loading the PSA solution, it is binding here, and then you're getting the signal output in this particular region, in the detection pad. So this is the another type of the system, as I said, LFA, where the paper has been utilized for the complete analysis. So for you all students, these four type of the pad is important: sample pad, conjugation pad, detection pad, and absorbent pad. Okay. Now another type of system is what? It's a micro pad, basically. Okay. So this is basically when the digital response has been, integrated. It means you have a components where you will be loading the sample, and after loading the sample, what happens? It will be converting system into the output in terms of the digital response. So this is the one of the another example which has been published in 2016 in analytical chemistry, which detect what the target compound, and due to this reaction, you are getting the output signal, which you can see in the digital form. So this is basically also called as a MEMS, which is known as a micro electromechanical system. Okay, so this is the third type of the system which has been utilized based on the paper-based matrix. Okay, so I have given the comparison here of the paper-based diagnostic system, like dipstick we have discussed, lateral flow assay, micropad. So if you see here, the possible detection mode in this case is what? Optical. It means the color. In case of the lateral flow assay, it is optical plus electrochemical. Micropad, it is optical, electrochemical, chemiluminescence, and MEMS. Right. I haven't mentioned the MEMS technology here, and I haven't mentioned the electrochemical system here, because this is the upgradation. If you can see, the possible detection mode is only 1 here, 2, it is 4 here, right? So that is what over the type of the system what you're developing, it gives you the advanced version, right? So of course there are some advantages of the system, there are some limitation of the system, but of course if you see the commercial volumes which is present in the market, every system has a commercial diagnostic system in the market, right? Except to the micropad system. So this we have recently published in 2017 all this data, and so in order to develop the paper-based system, you need to have the matrix first, right? The matrix which is very much important and commonly used is what? It's a Whatman paper, and everybody know the Whatman paper because they are using in the chemistry lab, in the biology lab, and so on. So basically, the paper, the number Whatman paper number 1, 4, and so on, 41. So these are the different, the range of the Whatman paper which is utilized based on the the pore size and the how much the sample is flowing and so on. So basically these are the things which is important to consider. And in addition to the Whatman paper system, the nitrocellulose membrane system is also being utilized for the development of various paper-based biosensors. Okay, so I will be showing few of the examples what we have developed in our lab. So one of the classical examples what we have developed in the lab is what? The paper-based system for the quick detection of alkaline phosphatase. Okay, so alkaline phosphatase is basically the milk protein. Okay, it's an enzyme, and in the raw milk, this enzyme is present. And when you go for the pasteurization process, this enzyme is denatured, and that how you will confirm the milk has been completely pasteurized or not. So this all whatever you are looking at this slide is what it has been done on a paper system. And what I've shown in the previous slide are Whatman paper. So we have used the Whatman paper here. So we have not developed a lateral flow. We have not developed a dipstick. We have not developed a micropad. This is a paper button system. Basically, we are calling it as a button. right? So in this case, what we have done basically, we have taken the Wattman paper. We are doing the punching system. You just use office punch machine, and you will get a disk out of that. And on this disk, these are the functional group which is present, and then you have to do a lot of wet chemistry on this, and eventually you are going to immobilize the antibody which is being selective for this particular antigen, which is a ALP in this case. So there will be a complexation reaction, and then you will be adding the BCIP chemical here. I'll be discussing in detail in the next slides how it works. And then there is a dimerization reaction, and eventually you're getting the color change. So this color change is also the optical system. It's not electrochemical system, right? So this optical system can give you the yes or no analysis, basically, whether the ALP is present in the milk or whether the ALP is not present in the milk, right? So now with this system, this has been integrated with the mobile phone, and the DIC analysis has been done. It means a digital image colorimetry, and then you are getting to have a complete calibration plot. So I have shown this type of the plot earlier also in the slide, where I mentioned the LOD, LOL, LOQ, and other variables. So these plot is very much important for any paper-based sensor or any sensor rather. So this shows that the paper matrix can be utilized for the complete development of any probes, any bioprobes, right? In this case, the ALP based system, okay? And this is a qualitative system here for a naked eye detection. But when it is integrated with the DIC, the digital image calorimetry, this is a quantitative system also, right? So here what has been done, this is the RGB wheel. Based on this RGB wheel analysis, we have calculated all the the values which is related for the on-site analysis of the ALP. So the innovation in this case is what? The waste paper disc has been used, and the low-cost device for affordable technology has been developed. In this case, students, we no need of any instruments. Just the color change will be enough for qualitative. For quantitative, of course, you need to have a mobile system and a complete data in that. And here we have also tested the system for in a commercial point of view, from in the village and from the various milk outlets. This system is extremely eco-friendly system, and we don't need of any complex procedures, and it can be operated by a common man. So this is the importance of the paper matrix where the sensor has been developed. So this is the construction part. You will take the paper system. This is the cellulose fiber in this case, and you have to activate the paper having the disonium system, and then you will going to get an activated paper. After this process, we'll have to have a EDC NHS treatment, which will give you eventually the immobilization of the antibody in this case, because if we don't have the immobilized antibody in this case or the bioreceptor, you are not going to have a selective system for the analysis of the alkaline phosphatase in this case. So after this process, what is important? You need to have what? The BCIP, as I said. So this BCIP is reacting with the ALP system, and eventually you are getting the dimerized form of this BCIP, and this is the colored product, basically this one, right? So this colored product can be visualized by naked eyes, as I have shown in the schematic representation. And after this process, what will happen? You will take the image and from the mobile, and you will do the processing, internal processing, and then you will be calibrating the system for the exact values of the ALP in the milk. Okay. So this is the final fabricated system. These are the very minute dimensions of the paper-based sensors, and you can see very clearly the size of the paper-based sensor. It is almost, very less than a 5 rupee Indian coin, and this is the final prototype which has been developed. With the increasing in the concentration of the ALP, the color intensity is increasing, and you can keep this in the wallet, the portability of the system, and you can see the weight. It's a 0.38 grams. So this is the first example of the DIC based protein analysis, and this is the very clear cut, the description how the system has been developed. So this shows, guys, the importance of a new type of the paper based approach, where neither we have used the lateral flow, we have not used the micropad. It's not a dipstick. This is simply the paper button, what we are calling. Okay, so basically not only this one, the system was further improvised, that the processing with the mobile system has been done externally. I mean, you need to take the image, then you have to process it in the laptop, and you have to do a lot of things. But after this process, what we have done, we have basically, integrated a software into the mobile. And then this is also we have done recently. We are going to apply for the patent of this one. So within the mobile, we have developed the app, and the whole process of the sensing based on the paper will be, integrated with the smartphone, and you are getting to have such kind of the response. So this is the one of the things what we are going to patent it very soon. So this will be kind of a control panel. You will have a control, you have a contaminated one, and you have to see the contaminated two. Which one is more contaminated? Based on the color, you will be knowing that. Okay. So, so what is the point now? Basically, the point is what? Sorry. So the paper-based system is going to give you what? Assured diagnosis. It means affordable, sensitive, specific, user-friendly, rapid, robust, equipment-free, and deliverable for the end user. So all things, whatever is required for assured system, it comply through the paper-based system. I have shown you that in the case of ALP, there was no equipment required for a qualitative analysis. It is extremely rapid, extremely robust, very sensitive system. It is very affordable. If you ask me the price of that, it is about 150 rupees maximum in the laboratory scale, or else if it is also deliverable. Basically, you can deliver it on-site analysis. So the complete, as per the WHO, right? The definition of the assured, lies in the paper-based system. So, okay. Now what is happening nowadays? So people are, using various external instrumentation, right, to enhance the performance. So these are the possibilities, basically. For example, the desktop scanner system, right, or a portable glucometer kind of a system, right, or you have to make a customized instrument, this one, or even the mobile phone. So whatever has been done externally, that can be integrated in this smart system, right, and the external instrumentation for the quantitative analysis. That's the point, basically, what we have shown in the next one, basically, where we have linked with the system with the mobile phone. So that is what I'm saying. These are the options. Basically, you can use either of this one to have the external instrumentation for smart analysis. So this one. So so basically, so this system is, these systems are very much important to integrate the mobile based system, right? Okay, so I will be giving few examples now where the paper microfluidic system has been utilized for the detection of PSA. Basically, it's a prostate specific antigen. These are the case studies. So this paper is already published in Analytical Chemistry from some other group. It's not my work. So basically, what is happening in this case, you can see there are multi-point where you can load the samples of various concentration. What is I'm trying to explain here? What that in the same chip you can load various concentrations, and with this you can have a calibration plot. For example, these are the ranges, right? And with these ranges, the calibration can be done. So it means not only for one compound, like in the earlier case I have discussed about ALP, you can go for a multi compound also. Why so? Because for one disease, you cannot go for one marker nowadays. You can go for a multiplexing system. So that is why this is very much important. Okay, so this is one of the example. Another example is what, where the aptamer system, which is very new bioreceptor system, okay, and this it has been utilized for the detection of what, ATP, and ATP is very much important for pathogen detection. So in this way, what is happening, basically, the aptamer has been immobilized onto the paper-based matrix, okay, and then onto this paper-based matrix, they have added what, a 3-amino-9-ethyl carbendazole system here, and then when the ATP is reacting with this one, this is going out because this aptamer is selective for ATP, and based on this one, this reaction, HRP with the AEC, this color has been changing. So this color is also related with the concentration of the system. You can see the concentration of the target. So based on this, the calibration has also been done. So these are the examples of the paper-based matrix, which is important for the analysis. Okay, so this is another type of system here again, where the paper matrix has been utilized. This is utilized for various type of cancer antigens. You can see 4 different cancer biomarker has been used: CEA, AFP, CA129, CA153, and so on. So basically, one system of made up of paper can be utilized for a detection of various different type of marker system. Okay. So this is one of the very classical paper, which is basically the detection of the DNA system that was published in Science. Basically, it's very classical paper published in 2000. So in this case, what is happening? So they have what a DNA probe present onto the paper system here, and this is the gold nanoparticle, which is another DNA. It is hybridizing here, and then what happens basically? This is linked with the gold nanoparticles, right? So what happens when you have this gold nanoparticle in the system? So it has a property, to from a silver plus it goes to silver 0 basically here through the mediation of hydroquinone, and you can see the black deposition onto this system. So based on this integrated system onto the paper matrix, you can calibrate how much target DNA has been involved here. So this is one of the very classical example where the paper has been used. It is about quite long back. So the conclusion, what I'm trying to discuss in this case is what paper-based technologies have been found great attention due to its capability for serving the demands. And the important thing is what the POCT device, point of care testing device, has been developed, and it is going to reduce the burden for the diagnosis. And the problem sometimes is what the analysis is sometimes semi-quantitative, right? So there is a still requirement for various calibration system in order to improve the sensitivity of the system, right? And sometimes what happens, the engineering system based on the various aspects, like how to design the minute the thing which is responsible for the whole development of the process, that has to be resolved in any case. So these are the future prospects and the conclusion, and these are the acknowledgements. I'd like to acknowledge my all students and people who have worked on the paper-based system is Kuldeep Mahato and other people. And this student has been recently joined my lab, and he's working on various aspects of development of mobile interface. So thank you so much. And if there is any questions or comments or any doubt, student can reach me at my email, which is pranjal.bce@iitbhu.ac.in. Thank you.
Can we ask the question right now if it's possible?
Hello?
Sir? Hello, sir.
Yes. Just a second. Yeah.
Yes, Gaurav.
Hello, sir.
Yeah. Please, Gaurav, tell me. Yes.
Thank you very much, sir, for such a wonderful and informative session. Sir, my question is that you talked about like paper bioanalyzer, that will be contamination detection. So can we use this paper bioanalyzer for the to check the heavy metal impurities in the soil?
Yeah.
Like, can we design this?
See, one thing is very important. Whether you are going for an optical analysis or else we are going for the electrochemical analysis. So the mechanism is very much important. Any target is able to be detected by paper-based system, either a soil contaminant or anything. So we need to know the reaction, what is happening basically. If there is some change in the color, if you give some specific, the target, then I can tell you how to tune that. Gaurav.
Yes, sir. Sir, suppose that if I'm preparing a nanoparticle and alongside that I am specifying a biomarker which will give color with the heavy metals that are present in the soil. So can we how can we prepare that in terms of this device? How can we prepare such system?
See, if you talk about some heavy metal, right? So if that heavy metal is reacting with some compound which is exclusively giving a color, right, then it is okay. But if it is not an exclusive reaction between the heavy metal or some of the compounds, then it will be difficult. Because for example, if you talk about lead or cadmium, if lead is giving the same interaction with the substrate, or if the cadmium is giving the same reaction, then the differentiation between lead and cadmium will be difficult. So that is the point. So you need to understand what is the reaction actually which is happening, because the reaction is very much important. But if, for example, if you are going for the electrochemical analysis, because all the signal, if you see in the voltammetric system, which is also the part of electrochemical analysis, is basically you're getting a signal at a particular voltage, basically, right? Potential. So if the potential is different of the heavy metal, then of course we can do this kind of analysis in the soil. That's not a problem. But my take home message is what? Yes, you can do that. That is possible.
So like in terms of electrochemical, like this kind of concept, if we are using, then can we detect the plant stress like while it is suffering from the viral load?
Right. See, I have given you one example where ATP was detected, right? Right?
Yes, sir.
Right. So in case of the plant stress, right, you see the point is what, in case of sensors, either electrochemical or paper based or any sensor, you need to know the marker first, right? If you are going to diagnose the plant stress, but you need to have some the compound or the situation, right? Or change in the temperature, or change in the pH, or change in the the any change in the plant, that has to be known, right? If we know that, of course, then we can think about the process by which you can develop the sensors. Do you understand what I'm trying to explain?
Yes.
If somebody is going to hospital, if somebody is a possible diabetic, what doctor will do? He will go for a glucose or he will go for a glycated hemoglobin, right? So they know the marker, right? So in the case of the plant, from any disease situation, if we know the marker that this concentration increase, for example, oxygen, gibberellin, or some, I don't know what exactly is happening in the plant system in case of the stress. If something is increasing, right, that can of course be handled.
Yes, sir.
Right.
Sir, I have an idea with respect to this.
Yes, please go ahead.
Sir, like when some kind of crop or plant is infected by the virus, so what a virus do? It generally attacks the protochlorophyllite oxidoreductase enzyme, which helps in the chlorophyll production. So when it is getting affected, so for sure the the green color of the plant will be decreased and it will produce yellow color in the leaves. But on the basis of initial detection, we need to prepare such kind of devices, because if we see the concept of plant viruses when they are affecting the plants, then it on the basis of initial stages, it goes very slowly because we can't detect the phenotypic expressions instantly.
Right.
And that's why the other crops also get infected on the time like we are detecting the main.
Right.
So we can work on such system like by focusing upon the POR reductase enzyme.
Right. I mean, that is possible. That is possible because see, the enzyme what we're talking, you mean that enzyme activity is increased, right? Something like that? Or
no, sir. Like its activity would be decreased.
Decreased, right?
Yes,
so
yes.
If the and what is the location of the enzyme?
Sir, location is inside the cell, like it's intracellular.
Okay, so that makes some sometime a little problem, because if it is intracellular, then you need to lyse the cell, get the enzyme out. We can check in the different stages of the infection what is the the enzyme, how the enzyme is behaving. So it would be better if something is other type of the marker, maybe something which is very much common, so
yes.
that can be integrated, right? Because I mean, I understand if you have a plant, within that system, if you have a marker, then you need to get the marker out and then you have to process it. I mean, the simplest example, if somebody is going for a urine analysis, giving a sample of urine is not a problem, but getting the sample from the kidney is a problem, right? So that is why even the kidney function test is been done by urine. So something like that, if I'm saying. So in the plant system, if something the marker is very easily accessible,
okay,
by some pre-treatment step or something, so that can be done. I mean, people have done a lot of even in the plant system. I'm not saying that people have not done. There are a lot of literature. If you will send me email, I can share you that details also of some papers. One of the Canadian group is also doing on the plant-based diagnosis for the plant, basically using the sensor system. Not only paper, other sensors also they have developed. So people are doing, but exactly you need to know the location of the marker, right? Or I mean, how you have to, I mean, if it is a very tedious job to get a marker out, then what is the point of going for the sensor? Because sensor is very robust system. It will not fall in the assured, what I said.
Yes.
Right.
Yes.
So we need to simplify, but if we are having a very complex system to get the sample out of that from the plant, this itself is a very tedious job, right?
Yes. Yes.
Right, Gaurav?
Thank you,
But
sir.
anyway, it's very good that you asked these questions. Nice.
Thank you, sir.
Yeah, very
So,
good. Yes.
so for the calibration techniques, as you mentioned before, there are
Who is
certain
this? Hitvi, right?
hit.
Yeah, yes,
Yes.
please,
For the
Hitvi. Go ahead. Yes,
techniques.
please.
So, is the sample retention rate or the retention rate a point for a low efficacy, like for paper-based techniques? And इसकी वजह से ही calibration techniques में problem होता है ऐसा कुछ?
नहीं, ऐसा नहीं. No, not like that. See, calibration system is an analytical, performance basically of any sensor. It is not only for paper. So if you go for HPLC also, that can be also linked with HPLC. But with reference to the paper-based system, this is a basic analytical performance parameters. So as I said in the example also, in case of glucose, if you are making the paper-based sensor which is going to detect glucose from 80 to 150 mg per dl, that sensor is not that it's not that good, right? So based on this calibration system, we try to improvise the system to make it more wide. The detection limit should be less, right? The linearity should be good, right? So that's how we focus. First step is the calibration. We have to keep this in mind that we need to make that sensor as good as possible in terms of the calibration variables, whatever is on the plot. I mentioned LOD, LOQ, LOL, and dynamic range, and so on, right? Is that what you asked, Hitvi?
Yes, absolutely.
Hello?
Thank you. Yes, sir.
Okay, all right.
Sir, good morning, sir. Am I audible?
Yes, Rishabh, please go ahead.
Yes, sir. I just wanted to ask you that the paper-based techniques which you have developed, sir, to check this, for the sterility check for milk, sir. I want to ask you what's the specificity and sensitivity of the test, sir?
So, okay. So, if I talk you with reference to the specificity first, right? So, I said that we have developed a ALP system by doing a lot of wet chemistry onto the paper buttons, right, which was coming from the punching machine. After the process, we have immobilized the antibody, right? Okay, so that antibody is specific towards the alkaline phosphatase, which is present in the milk. Okay, so that is why we want to make a system, specific. Okay, because the antibody is there, it is not going to interact with some other things. So that is a standard, concept of immunology. So based on the antibody, the system is extremely, specific towards this system. Okay, and another thing is what I have not shown all the data in this one. If you will go to that paper in detail, so we have done a lot of control experiment with antibody, without antibody, with BCIP, without BCIP, all permutation combination we have tested, so that we should not get a false positive results, right? So that has been done in very much detail in the paper. So if you see that, you please go ahead with that paper. If you don't get that paper, please write me email, I'll send you. Okay, so this is because the system is very specific. Now, this is the first question, what you asked. The second is what the sensitivity, right? The sensitivity system is very good because we have calibrated the system in a way that after doing a lot of optimization reaction pressure, right? So that system works very well in the range where ALP is present naturally in the milk, right? And when you do the pasteurization, the ALP is denatured because it is enzyme, but the temperature of denaturation is quite high for ALP. That is why it has been used as a marker system for pasteurization. If you go in any industry, mother dairy or Amul or and any milk industry, they have used this system, right? Not ours, ALP system, right? For the to check the system, right? So in terms of sensitivity, it is highly sensitive, right? So that's not a problem. So all the dynamic range is very wide. If you see the response of that one, what I've shown in the color chart, what I have mentioned, protein detection kit, it goes from 10 unit per ml to 1000 unit per ml, right? So in this range, basically, all the values is coming with reference to the level of ALP. Okay, Rishabh? Hello?
So do we have any numerical data? Like, for example, if I talk about specificity and sensitivity, we say it's like if I talk about lateral flow, so we say it's like the sensitivity level will be around 98% or 99%.
Yeah, of
Such
course,
kind of data.
there is. So I'll tell you again. With reference to the specificity, right? How much it is specific, we go for the analysis of specificity coefficient, case select, we call it case select. So I have not mentioned, Rishabh, in the paper. So we calculate the coefficient values, how much it is selective, right? So that is, of course, we are doing that, right? And in terms of the recovery, in terms of how much it is specific in the milk sample, we go for a percentage analysis. That in the see, when you develop any paper matrix or paper sensor, you have to calibrate that system first in the standard buffer, right? Okay, and then with that all optimized system, you have to go for in the milk system, right? So how the system is behaving in the buffer, and after moving into the in a milk system, how it is behaving. So that system is calculated by having the equation percentage recovery is equal to, you have to put the numerical values with how much you are getting in a buffer and how much you are getting into the milk sample. Then you have to multiply by 100 and you will get a percentage how much recovery is there. This is one way. Another way is, there is another method is called a standard addition method. So by that you will know how much concentration is there. So all what is you are mentioning, it is mentioned very much detail in the paper. So you please go to that paper. If you don't get that paper, please send me an email. I'll share that paper with you. Okay, Rishabh?
Yes, sir. Thank you, sir.
Yeah.
Hello, sir. Good morning.
Good morning.
Subrajit here, sir.
Yes, Subhajit,
Sir,
please tell me.
Sir, I have a question regarding that ALP biosensor, alkaline
Yes,
phosphate biosensor.
Okay.
Sir, you are using this biosensor to detect the milk only. Only you are using it only milk, na detection. Sir, did you know, sir, there is a if high amount of alkyne phosphate, there is a ALP test in biomedical there is a device which is used to detect bone degradation as well as gallbladder and liver problems, sir. Sir, have you ever tried your biosensor to detect alkyne phosphate in blood?
Yes, yes. Not in blood. We have done in serum.
Serum.
Right.
So,
Yes. So
what type of result you have got?
So, see, just a second. I'll show you that paper. Just hold on. Dr. Megha, we have enough time, right?
Yes, sir, up to 11:30. Then we have next session. So
okay, no problem. Let me answer these very good questions. Just give me a second. I'll share the slides. Okay. Shubhajit, just one thing. The paper-based system what we have developed, right? We have we have tested that in diluted serum first thing. We have not reported so far. But we have developed one more sensor which is also for ALP, right? Just give me a second. I'll share that paper with you so that you will understand. Just a second there. Because it's better to show that and then, you know. Rojit, can you see this paper, please?
Yes, sir. Yes, sir.
Okay, right. So see, and this one, another one.
Yes, sir.
Paper-based immunosensor naked eye ALP detection, right? You can see this one also.
Sir.
Yeah. What Rishabh? Just I'll take one minute to explain Rishabh. Rishabh, can you listen to me? Hello.
Yes, sir. Yes, sir.
Yeah. What Rishabh asked basically, these are the values, right? These are the selectivity assay, what we have done. These are the numerical values basically, right? So based on this one, we have done this particular analysis, okay? how much these are the interfering molecules, right? This is the ALP signal, so we are not getting any color. So these values are coming because of this particular calculation, right? And after this one, the recovery one, this part. So how much, when you're adding the ALP in the milk, how much the recovery percentage is there, right? So this is the response of ALP, right? In the spikes, and this is in the blank system. This is the this is the ALP in the analytical response in the standard solution. So based on this one, we are getting these values, right? So you can see very clearly. I have not put this data in the presentation. So this is the buffer data, right? The red one. So, and this is the milk basically. So in the milk, when you do this one, you are getting very close response. It means this works very well, right? And these are the percentage value, right? This one, right? The sensitivity was 98%, right? Up to 18 days or something like that. So I mean, all these details are there in the manuscript, right? So we can go through that. Now I'm coming to Shubhaji questions, right? Now Shubhaji, can you see this slide?
Yes, sir.
In serum alkaline phosphatase, right? Okay, so see this, the earlier one we have tested in milk and also in the buffer, in the serum, diluted serum, right? But this is also the alkaline phosphatase. This is, I have not mentioned because that is not a paper matrix, right? So basically in this case, if you see, this is the screen printed electrode, right? On this day, in this case, we have done a lot of, the nanomaterials application. After this process, we have, loaded the antibody, which is for the ALP, right? In the earlier case, we have utilized the BCIP, okay, and then we are getting the color change. However, in this case, we are studying the resistance of the surface. Resistance of the surface means what? This is the electronic chip, right? When you're loading these samples, the electron transfer because of these materials, if you can see my moving cursor, students, this is increasing, right? Because these are the nanomaterial. However, when you're loading this protein, this Y-shaped antibody and this ALP target, the resistance is increasing, right? It means electron transfer is dipped. So this concept was utilized in this chip basically, and then we have taken a clinical serum sample from the hospitals, right? And we have tested this ALP in the sample, which has been done, which is obtained from the clinic, right? So this is the data what I am mentioning here. So this is the classical version what I mentioned in the previous case here, the ALP one, and then we are increasing the concentration ALP here. I can see the concentration of ALP is increasing here, and the signal is keep on increasing here. That's what, right? So what I'm trying to say that with this system we have also tested, right, this one, but since the dynamic range in this case in the serum, because of some other reason, we don't know that, so that was not that very good, so we moved on to the electronic system, right? Okay, so yes, Subhajit, that we have done all, of course.
Yes, sir.
How
Yes, sir.
the sample in the serum which can be tested, right? So we have done all the analysis, and you can see in the blood serum, in the PBS, right? You can see that. So
Sir, in the electronic,
sorry, what?
device,
Yeah,
please show that slide, sir.
Mhm.
That in
Right,
the electronic device where you have used it as a ALP detection, what type of result you have got over there? Means what is the means sensitivity over there in
The
electronic
sensitivity
device?
is very good. You can see it here. You
Sir,
can see it.
sir,
Yes.
means sir, it may be sir something monitoring problem, or there is signal analysis problem that you need to recover. I means if sir the sensitivity is good well enough, then
Mhm.
it must work, sir. Because I am also working on a same device that is a glucometer type. Due to some means a device cannot work due to some signal analytic process. Means if the analysis is not going on in a perfect way, or the monitoring process, if the maybe the biosensor is working well and good, but the analytic process is not good in a means electronic means. So that's why the result may be different, sir. Because this system can be a very good tool, means in rural area where people can
Right.
detect a liver problem,
Right.
gallbladder problem, as well as bone degradation problems.
Right. See, so one more thing I would like to add in your discussion is what, when we optimize this system in the standard, right, in a buffer. I mean, you can understand this very well now because I know that you are working in this area. But when you go for the samples, right, real samples like a blood or a serum, or anything, there is a matrix effect. So that matrix effect is very detrimental. So the system analytics is fine, But the major problem is coming because of the sample interference, which is an unknown, very complex matrix. That is technically called as a matrix effect for any sensing system, right? So for that reason, there is a model, right? The standard addition, because you need to dilute the sample, and then in that diluted matrix, when you are testing that, then it will be okay. And then where that dilution factor you need to multiply and you have to do the back calculation, then you will know exact concentration what is there. So system analytics is okay in the standard buffer, but when you reach to the standard samples, that makes a problem sometimes. That is why there are, for example, 1000 sensor developed, maybe 1 or 2 they come in the market. That is a problem basically.
Yes, sir. Yes, sir.
Right. So it's very good that you asked this question. Very nice. Okay. All
Okay,
right.
sir. Thank you, sir.
Yeah, no problem.
Any more questions?
Hello, sir.
Yeah.
Sir, like for colorimetric analysis, sometimes we use sir mobile app to analyze. So, like there are different camera which take different photos, sir. So that different colorimetric result can come. So, sir, how can we overcome that?
See, this is a very, very good question. The light, what type of light you are using, right, in the whole analysis, right? That has to be optimized, right? Whatever, I mean, we have also done this one, right? If you have seen that. So we have done,
Yes,
we
sir.
have, I mean, again, I'm telling you, we have mentioned this in our papers also, right? That this is a light condition, okay? So the light condition, the camera type, the pixels, values, and everything we mentioned in the manuscripts, right? But how to overcome this is what basically you need to do the whole analysis for different type of systems, right? And then you can conclude with that in this system it work like this. But for all system it is not possible to do that. So I am just sharing the details again here. Just a second. You guys are asking very good questions. I'm very happy. Right. Divijay. Sorry. Divyansh Yadav, right?
Yes.
Right. So Divyansh, you see this manuscript, right? What this is the system, right? You see
Yes.
this details of smartphone specification. We have mentioned this, right? Okay. Hello.
Yes, sir.
Right. So that's what I'm saying. So this we have to mention in every case. What is the the light source, what is the smartphone system, what kind of the we are using. But as I said, in a particular type of the camera, right? The sensitivity of the system also, of the camera system, the light, exposure time, everything is important, right? When you do this kind of application, okay? But in general, you can develop the application, okay? But it is not like if you are getting a response of 2, for example, but with other system, you are going to get 200 response. It is not there. So that is why you can, do the analysis for different type of the camera system, right? And then you can take a, conclusive data that how it is working in all the system, right? And you can put the error bars and how the deflection is there with reference to, different type of camera system, right? Okay.
Sir, for to overcome, can we do the calibrating thing? Sir, means we can click a normal photo without the any color, means to just see the environment, it is dark or something, then we can calibrate it.
See, calibration you will be doing in any case, right?
Yes,
Without
sir.
calibration, you will not be having the analytical, performance of the system. That is not possible. So what I will comment on this, what you need to, do the analysis with various camera, right? And
Yes,
then
sir.
you can take the average of that. That average values can be used for the calibration system, right?
Yes, sir. Yes, sir.
Moreover, if you see some other papers, sometimes they mention the data in grayscale, right? Instead, the exact color. Like I have mentioned the gray, this one, the green color, right? But some people they mention in the grayscale. So that is also, maybe linked with that particular type of the problems. But of course, with other system, you can, make the all range of analysis and you can make the mean of that. Then you can make the calibration based on the mean values. And then you will have a, error values. So all the data will fit in that basically in the calibration range. Okay.
Okay, sir. Thank you, sir.
Yeah. Hello.
Any more questions, students?
Yes, ma'am.
Yeah, please go ahead.
Sir, can you please explain the aptamer's principle again?
Akanksha, right?
Payal.
Oh, sorry, Payal Bansal. Okay. So Payal, so aptamer is basically, it's a type of bioreceptor. Okay. So if I say the typical definition of aptamer is what? The aptamer is made up of a word aptus, right? It's a Latin word. It means to grab. What happens if you can see my hand? So this works at a principle of target induced conformational change. So if it is a target, if it is aptamer, aptamer is nothing, it's a nucleotide sequence. Okay. So it is standing in a normal position, but when some target comes like this, it makes a conformational change, like something like this. So that is why I'm saying it is in the position, but when the target comes, it changes the position. It makes a quadruplex, right? So that quadruplex, because of that, the molecules, retain in the system, right? So that is one of the, the classical definition of the aptamer. And as I said, it is made up of nucleotide. So that is the mechanism through which it works. So it is different from the antibodies, right? It's not a protein, right? So that is why people are using a lot aptamers nowadays, because of its the stability system, right? It is able to synthesize. So that is why aptamer is very powerful bioreceptor nowadays. And with reference to that paper, what I mentioned with reference to ATP is what? So since aptamer is made up of nucleotide, you can have a complementary hybridization, either full or even half, right? So what they have done, basically, they have taken a HRP system, right? They have hybridized with the aptamer partially, and when the ATP is coming, what is happening? The hybridized part is out, and aptamer is grabbing the ATP. As I said, target induced conformational change, right? And then there is a biological reaction, and then the color is changing because of the particular substrate which was pasted onto the paper disc. So that is why it was happening in that particular paper, what I mentioned with reference to the ATP detection. Okay, Payal, is it clear?
Yes, sir. Sir, ये किस diseases में work करता है?
Aptamers.
Yes, sir.
See, aptamer is basically used for various. It doesn't work like in a case of disease. So, I mean, it's a bioreceptor. If you read a literature, you will find a lot of sensors basically where the aptamer has been designed. So, for example, cancer biomarkers, drugs, even whole cell bacteria, staphylococcus, downomycin. We have. I mean, I have also done a lot on aptamers, downomycin, kanamycin, sulfonamides, various type of targets, from small molecules, not small molecules, even the metals, heavy metals, to small molecules like drugs, till the peptides, the proteins. For example, the cancer biomarkers, HER2 is a breast cancer antigen. We have done that also. Other people have done for CEA, PSA, right? And the whole cell, from the smallest metal ion to the biggest cell. So that is possible with the aptamer. Not only for diagnosis, this is also used for therapeutic, like drug delivery system also. So that is the the advantage of aptamer, or the application of the aptamer in the diagnostics. Okay, Payal?
Okay, sir. Thank you.
No problem.
Anyone else wants to ask? So I think there are no more questions. Thank you so much, sir, for your time, and
Yes.
your sessions are always wonderful and informative. But I am very happy that students are interacted this way. They're getting interesting in.
That is very important actually, Dr. Mega. I am very thankful to you for sharing the Excel sheet actually, because I wanted to make the presentation that students should understand that, right? It is not a platform that we should come out and speak a very high level of research or the people, the student, who are participating in the whole process, right? They should be benefited. That is our task.
Yes, sir. Yes.
Right. So that is very, very good. I mean, I'm very grateful to you for sharing that detail, because I was in a dilemma. I mean, I think in December or somewhere October, I also talked in the 6th summer school.
Yes,
So
sir.
that time I was not knowing the the audience, the type. But this time you have helped me a lot. I'm very thankful to you for sharing that details. So I've tuned the slides in that way so that people will understand.
Yes.
Right. Yeah. And I'm also very happy that students have discussed so many things and very curious questions and very technical questions even they have asked. This is very good actually.
Yes, the students are brilliant. Actually, we got.
Yeah, very good, very good. I've seen. I mean, most of the people they're coming from very good institutes, right?
Yes.
They have given some proposals and sort. I've read that Excel sheet. Yeah, thank you.
Thank you once again, sir. And we look forward for your contribution and support for the workshop.
Yeah.
Thank you, sir.
Thank you, Dr. Man. Thank you. Okay, bye, students. Bye.
Bye, sir.